Yeah, hi there everyone. This is Ashmita from Saurabh's classes. We are going to solve a bunch of questions from the IIT JAM Biotechnology question paper of 2023. Let's start with question number 22. It says match the microorganisms in group 1 with the human disease in group 2. Group 1 and group 2. This is group 2 and this is group 1. Let's try to understand which one will go with which one. Treponema pallidum. This one will go with, yes, this one will go with syphilis. So syphilis is in 4. Next is Trypanosoma cruzi. This one will go with Chagas disease. So Q with 3. Trypanosoma gambiens. This will go with um, this is actually sleeping sickness. So this will go with sleeping sickness. R1. Bordetella pertussis. This will go with, oh, which one is left actually? Whooping cough. Yeah, whooping cough. S2. Now let's go through the options. Which one is similar to this one? Oh, it's in option one, A only. So option A is your correct answer. All right. Let's move over to the next question. It says, which one of the following is correct in the case of conjugation of a high frequency combination strain with the strain of E. coli? In case of conjugation between a high frequency recombination strain and an F strain of E. coli, we can say the correct statement would be recombination frequency is low, F factor transfer frequency is high. Only then this would be correct. Okay, in this case it would be correct. Now during conjugation, what happens? The HFR strain, it transfers the chromosomal DNA to the F recipient cell, and the recipient cell, what does it do? It can become partially diploid. Okay. However, since the transfer of chromosomal DNA is a slow process, hence we can say the frequency of recombination events is also slow. On the other hand, if I say the F factor is present on a plasmid, then if present on a plasmid which can be transferred at a higher frequency than the chromosomal DNA, then the frequency of transfer of the F factor is high. In that case, F factor frequency would be high. Let's move over to the next question. That is question 24, and it says there is a reason and there is an assertion for it. Assertion and then there is a reason. Determine the correctness or otherwise of the following assertion and the reason. Assertion says the cardiovascular organization called double circulation provides vigorous flow of blood to the brain, to the muscles, and to the other organs. And the reason says the blood is pumped a second time after it loses pressure in the capillary beds of the lungs or skin. Now, here I might say that the assertion is true, but the reason for this particular assertion, it's just not going. It's not the correct explanation, I would say like that. So let's not go with the reason. So we can say that it's actually this is correct, but it's not the correct explanation for the assertion. Uh, if we just look at the reason individually, that's correct. So let us go with option B. Both A and R, that is assertion and reasoning, are true, but the reason is not the correct reason for the assertion. Okay, that is what number B is saying, and hence we are going with number B. Now, talking about this in a little bit detailed manner, I can say that double circulation, okay, double circulation, the term that has been used over here, double circulation is a cardiovascular organization that involves the circulation of blood twice through our heart for each complete circuit of the body. Okay, that is what double circulation means. And the first circulation here, as we know, there are two circulations taking place. The first circulation is called pulmonary circulation, where the blood is pumped to the lungs to pick up the oxygen and then release the carbon dioxide. On the other hand, the second circulation is called systemic circulation. So first one is called pulmonary circulation. Second one is called systemic circulation. Okay. Now, in systemic circulation, what happens? The oxygen-rich blood is pumped to the rest of the body to provide oxygen and nutrients, and also it helps to remove the waste products from our body. Okay. So that is how we can conclude that what double circulation is, and our correct answer is option B. Let's talk about the reason a little bit. Now, what reason is given? How is it correct and all? We are going to discuss it now. Okay. Now, the reason mentions that the blood is pumped a second time after it loses pressure in the capillary beds of the lungs or skin, right? That is written. But it is not the correct reason for the vigorous flow of blood to the brain, to the muscles, and to the other organs. Okay. The reason why double circulation provides a vigorous flow of blood to the brain, to the muscles, and to the other organs, it's only because it allows the heart to pump oxygen. It allows the heart to pump oxygen-rich blood to the body at a higher pressure, which allows the blood to reach these organs more quickly and more efficiently. Okay, so it is only because the heart is allowing. Okay, therefore we can say that the assertion is true. um, that is, it is true because the double circulation does provide a vigorous flow of blood to the brain, muscles, and other organs, but r or the reason is not the correct explanation for this phenomenon. Okay, let's move over to the next question now. We are focusing on question 26. A genetic linkage map represents what? Let's go through the options. Relative locations of genes on a chromosome, distribution of mutational hotspots, phylogenetic linkage among organisms, accurate physical distances among loci. Here, your correct answer would be the relative locations of genes on a chromosome. Genetic linkage map represents this thing. Okay. So, what is a genetic linkage map now? A genetic linkage map, uh, it shows the relative positions or distances between genes on a chromosome. These maps are mostly created by analyzing the frequency of recombination between linked genes, where the genes that are closer together are less likely to be separated during recombination than those that are farther apart. The map units which are used for genetic linkage maps, they are centimorgans, like this we write in short form, centimorgans, and represent the frequency of recombination between two linked genes. Okay, so we can say option B is incorrect because the genetic linkage maps are not used to show the distribution of the mutational hotspots, but rather the genetic linkage map is used to show the relative positions of genes on a chromosome. Okay, that is why the second one is incorrect. Now the third one is also incorrect. It is because uh, genetic linkage maps that I am talking about is or are not used to show the phylogenetic linkage among organisms, which refers to the evolutionary relationships between the different species or between the different groups of organisms. That is why C is also wrong. The option D is also incorrect because the genetic linkage maps do not represent accurate physical distances among the loci, okay, but rather it shows the relative distances or positions of genes on a chromosome. So physical maps, on the other hand, they show the precise physical distances between the loci, okay. So we have done a lot of discussion on genetic linkage map. So let us move over to the next question. Class 2 MHC molecules are not expressed by which one? It is not expressed by T cells. Okay. Now talking about class 2 MHC molecules. Okay. Class 2 major histocompatibility complex. That's the full form. Major histocompatibility complex. That is what the full form is. These molecules are cell surface proteins that present the peptides which are derived from extracellular pathogens to CD4 plus T helper cells. Okay, and these molecules are expressed on antigen presenting cells, which include B cells also, then dendritic cells also, then macrophages also. Okay. Now, option D is the correct answer because the T cells do not express the class 2 MHC molecules. Okay. Instead, the T cells, what do they express? They express the class 1 MHC molecules. Okay. And what do class 1 MHC molecules present? They present the peptides which are derived from the intracellular pathogens to CD8 plus cytotoxic T cells. Okay, so that was our discussion with this question. Now let us move over to the next question. It says, which one of the following enzymes is required to ensure the replication of a negative sense or negative strand RNA virus? Here, let us go through the options. No. Your correct answer would be RNA dependent RNA polymerase. Okay. Explanation would be like hmm, negative sense or negative strand RNA viruses, if I say. They have a single stranded RNA genome that is complementary to the viral mRNA. Okay. This means that the viral RNA cannot be used directly as a template for translation or for replication. In order to replicate the viral genome, these viruses encode their own RNA-dependent RNA polymerase. That is, we call that RDRP enzyme. Okay, this RDRP enzyme, can use the negative sense RNA as a template to synthesize the new viral RNA strands that can then be used for the translation or for further replication. Okay, that is why I have chosen option D to be the correct answer. Now let us discuss about the other options, why they are not correct over here. Let's start with option A, that is DNA dependent RNA polymerase. It is incorrect because the DNA dependent RNA polymerase, it's an enzyme that synthesizes the RNA. Okay, that synthesizes the RNA from a DNA template. But the negative sense RNA viruses do not have a DNA template. Okay. Next, talking about DNA polymerase. DNA polymerase is incorrect because over here the DNA polymerase is an enzyme that synthesizes DNA from a DNA template, and it is not involved in the replication of RNA viruses. That is why second option is also incorrect. Let's talk about the third option, that is option C, RNA dependent DNA polymerase. This is also wrong because the RNA dependent DNA polymerase, it is also known as the reverse transcriptase transcriptase. Okay, it is an enzyme that synthesizes the DNA from an RNA template. And this enzyme is found in retroviruses, which are a different type of RNA virus that have a positive sense RNA genome. Okay, that is why this is also not coming under the negative sense or negative strand RNA virus. Okay. Now let's move over to the last question for today's class. That is, let us match the recombinant DNA products in group 1 with its application in group 2. This side, the letter side is group 1 and the group 2 side is the numbering part. It's not given in proper columns. Let us try to group them together. Starting with the, tissue plasminogen activator, it will go with um, I feel the options are absolutely correctly placed. I don't need to change any of the options over here. Yes. Let's talk about this in details because it's already correctly placed. We don't need to change anything over here. P will go with 1, Q will go with 2, R will go with 3, and S will go with 4. So it's already correctly placed. Tissue plasminogen activator. This is a protein. Okay, this is a protein that dissolves the blood clots, and it is used for emergency treatment of the heart attack. Okay, so it is definitely correct because it has been matched or correctly placed with emergency treatment of heart attack. Okay. Let's talk about the second option, that's Q, erythropoietin. Okay, and erythropoietin is a hormone. It's a hormone that stimulates the production of red blood cells or RBCs, and it is used for the treatment of anemia. So it is already correctly placed. It is used for treatment of anemia. Let's move on to R, that is superoxide dismutase. Superoxide dismutase is an enzyme. Okay, it's an enzyme that prevents the tissue damage caused by reactive oxygen species, and this superoxide dimutase, it's used for it's used to treat conditions such as arthritis or inflammation. Okay, that is why it's written prevents tissue damage, so it's absolutely correct. Talking about the last option, interferon. Interferon is a protein. Okay, and this protein stimulates the cells to inhibit the viral replication, and it is used to treat viral infections. So here, okay, viral infection examples like hepatitis B and hepatitis C, and certain types of cancer also you can include under this. So it's already correctly written over here. So I would go with option A. I think it's the option A only that is correct because at first only we can see P1, Q2, R3, S4. Yes, go with option A. Alright, that was the discussion for today's class. If you want to have more active discussion with the teachers over here in class in live sessions, then you can join our classes for the better preparation of any kind of examination that you are going to give. If you are preparing for any kind of competitive examination or any entrance examination, then definitely you can join our classes for live sessions, for recorded lectures, for mock tests, for doubt clearing sessions, as well as for interactive discussions. So yes, that was it for today's class. We are going to meet again in our next classes. Thank you for watching.